So as I was saying, it's a pleasure after many years when we've wanted to work together to have an opportunity to do that. Now, what I'm going to talk about today actually has nothing to do with the collaboration that we're about to enter into. I had no idea when I was going to give this talk that it was going to be for this wonderful occasion. My congratulations to you all for having put together such a successful program for so many years. I think you've done an incredibly good job. But what I'm going to do is I'm going to tell you about a small story, a research project that my group has been involved in back at the University of Nottingham for a few years, which I hope will illustrate many of the points that have been mentioned already about the requirement, if you're going to do good medicinal chemistry, you can't just do medicinal chemistry. It's not just chemistry. You need the biology. You need all sorts of other disciplines. You need structures. You need all sorts of other things around you to make a project work. And the project that I'll be talking to you about is one, it's still very much under development. To be honest with you, we're a little bit stuck at the moment with the project because actually there's some things that we need for the next stage, some pieces for our jigsaw puzzle that we don't really have quite now. But I can tell you where it's got to so far, and I hope it'll illustrate how powerful it can be when you're able to get biologists and chemists and pharmacologists to all talk to each other and agree about what it is that should be done to try and solve a problem, a disease. So obviously, the disease in this case is cancer rather than a neglected tropical disease. Cancer is where I've done most of my work up to now. But all the principles, I think, are applicable. It doesn't matter what sort of medicinal chemistry you're doing. Yes, I thought I ought to start by explaining it is where I work. So I say I'm at the University of Nottingham. So this is this small island, which is the UK. And I think actually if we just zoom in the middle, Nottingham is pretty much right in the middle. So if we just zoom in a little bit and get really in a bit close, whoa, there it is. And so this is where I work. This building here is actually our local hospital. Well, this is the hospital, in fact, for the city of Nottingham. And this is one of the big sort of roads that goes around the edge of the city centre. There's a little bridge across it. And on the other side of that bridge, this building here, this is where I work. So this is the Centre for Biomolecular Sciences at the University of Nottingham. It's a research institute within the university. It's not part of one school or another. It's not part of chemistry or pharmacy or anything like that. But it's a building in which researchers, no matter which school of the university they come from, they can work in there if their interest is in biomolecular structure, basically. So this is where I work. I am actually in a school of pharmacy, although I myself am not a pharmacist. I'm actually, originally, once upon a time, I was a synthetic organic chemist. But this is where I work now. So that's not the prettiest picture. The University of Nottingham is actually a very green and pleasant place. This is the old centre of the university. This is when the university was first founded. It was a beautiful building. Oh. So the university is about 100 years old. It's not very old by UK standards. Originally, it was this rather nice building on this small hill beside a lake. Nowadays, it's much more developed. By UK standards, it's a medium-sized university, about 35,000 students. We're quite unusual, though, amongst UK universities in that we're quite international. We have a campus in Malaysia. We have a campus of the university. In fact, we have two campuses of the university in China. They're just campuses of the university. They just are a little bit further away than our other campuses. As I say, we have about 35,000 students, and it's a university that studies everything from archaeology, anthropology, and ancient Greek through to biomolecular sciences. It's a general university. But now let's go and talk about the subject. So let me start by introducing to you telomeres, or telomeres. So what telomeres are the structures that are found at the ends of our chromosomes. So every single one of our chromosomes, of course, is a length of DNA, and that DNA has to eventually come to an end. And the ends of our DNA are very special places. For a start, an end of anything is sort of a dangerous place to be. It's fragile, it's exposed. It's where nasty things can happen, and you don't want that to happen. So the ends of all our chromosomes, our telomeres, are protected. And they're protected by having a coat of proteins around them. And this coat of proteins together is called shelterin. In fact, you can see this is a metaphase spread of some chromosomes, and just at the very tips, you see the little green lights there? That's fluorescent antibodies to proteins in shelterin. So you can see the way that the proteins that are present in these regions are very, very specific. Their role is just to bind to this region of the DNA, not to any other part of the DNA, and to stabilize it. So shelterin is actually a mixture of at least 6 different proteins, obviously many copies of each. So let me just introduce them to you very quickly. So the first 2 are TRF1 and TRF2, and they are proteins which bind directly to the double-stranded DNA at this end of the chromosome. And the reason they do this, and they bind just in this region and not anywhere else, is because they have evolved to recognize very specifically the DNA sequence which is found in this telomeric region. Because this telomeric region is not normal coding DNA. There are no genes here. This DNA does not code for any proteins. It has a very simple repetitive sequence. It goes TTA, GGG, TTA, GGG, TTA, GGG for thousands of times. But that is the signal that we are at the end of a chromosome, and it is that sequence, TTA, GGG, which these proteins, TRF1 and TRF2, have been designed to recognize. So they are what basically localize this shelterin complex uniquely to the end of the DNA. There is also another protein whose job is to recognize telomeric DNA, and that's called POT1. As you know, that's the one we're going to be talking about more in a minute. So POT1 is different from TRF1 and TRF2 because TRF1 and TRF2 are designed to bind to recognize double-stranded DNA. And of course, as you know, most of our cellular DNA is double-stranded. But at the very ends, again, of our DNA, in these telomere regions, they're not. There is a single-stranded region where the sequence TTA-GGG, TTA-GGG extends without its complementary base strand of bases. And this region is recognized specifically by this protein POT1. Then there are 3 other proteins, TIN2, TPP1, and RAP1, and they are, if you like, the glue proteins. Then there are these glue proteins that knit everything together to make something which some people call like a bootlace. If you think about your shoelaces on your shoes, at the ends there you have these little bits to protect them, and that's a bit like what shelterin is. It's holding the whole thing together. So why are we interested in this thing? Well, what's interesting about the telomere is what happens to the telomere when a cell divides. Because just like the rest of all the DNA in our cells, it has to be replicated. And there's a problem. When our cells replicate a strand of DNA, or I should say a whole chromosome, we know there's a complex machinery to do that. This complex machinery is incredibly efficient at reproducing the sequence of all the DNA in all our chromosomes to create the copies of it, except when it comes to the very tips, the very ends of our chromosomes. Because basically the machinery can't read this far. It just stops. It's a bit as if you were photocopying a book, but you could never photocopy the first page or the last page. Now clearly, you imagine a cell just dividing and dividing. Imagine what would happen if you had a book, your favorite book, And every time you made a copy of it to give to your friends, one more page was lost from the beginning, and one more page was lost for the end. This would be soon a pretty bad thing. And you can imagine that if that happened to our cells, that would also be a pretty bad thing. Pretty soon we would be running out of some genes. Now that doesn't happen. It doesn't happen for two reasons. Firstly, that's why we have telomeres. Remember, the telomere sequence is just TTA, GGG, TTA, GGG. Just repeats. It's a bit as if you had a book and the first page of your book just said blah, 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 blah. Now, if you happen to tear that page out of the book or fail to copy it when you photocopy it, so what? The story in your book is safe because it wasn't on the first page and it wasn't on the last page. And that is part of the reason that we have our telomeres. Because if our cells divide and we lose a little bit of telomeric DNA, it doesn't matter. Our genes are still there. But clearly you can see that if a cell divided forever, we would still run into a problem because we haven't got a million pages of blah, 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 blah. In the end, we would erode our telomeres and we would start basically failing to replicate our genes. So the reason that doesn't happen normally is that normal cells, of course, do not divide forever. They replicate maybe a few times, but then they enter what's known as replicative senescence. Most of the cells in our body are not dividing, unless they're stem cells. Why is that? Because they know that they shouldn't really, because if they divided too many times, we would run out of telomeres. Though there's an automatic signaling mechanism that senses when telomeres get to a certain length and stops them getting any shorter. It's when our cells stop dividing. But what about if you're a cancer cell? One of the hallmarks of cancer is replicative immortality. You can replicate forever. How can a cancer cell replicate forever if every time it divides, it loses parts of its telomeres? Well, the answer is that since cancer is such a terribly clever disease, it has a protein called telomerase. It's a reverse transcriptase. And what telomerase does is every time a cancer cell divides and its telomeres get shorter, telomerase comes along and as a reverse transcriptase, it adds further repeats of the telomere sequence back onto the end of the telomere. So the telomere shortens when the cell divides and then is extended again by the presence of telomerase such that a cancer cell can divide forever. Now there's something else I want to tell you about telomeres and telomerase and the way in which they replicate. And that's to do with this single-stranded region here at the end. As I said, it's got this very specific sequence, TTA, GGG, TTA, GGG. We can draw it in this extended sequence, but of course that's not the shape that it adopts naturally. What happens to these G-rich sequences with Gs, 3 Gs in a row, is it will fold up on itself and very rapidly forms these so-called G strands or G quadruplex sequences in which 4 guanine bases hydrogen bond in a cyclic way, a lovely hydrogen bonded cycle, to form these folded up topologies. So really we shouldn't draw our strands looking like this, we should draw them something looking like this, all folded up on themselves. So they have a very specific structure with these guanine tetrads. The loops around them can be lots of different shapes, but this is their basic structure. So why is this interesting? Here's our shelter-in complex again, the one we were talking about earlier. Here's POT1 binding to our single-stranded DNA. Here's TRF1 and TRF2 binding to our double-stranded DNA. Here are the proteins RAP1, TIN2, and TPP1 that glue the whole thing together. This is the complex which is present at the end of our chromosomes. And we've said this is really important because if it breaks down, we expose the ends of our DNA. Things can happen, damage can occur, the cell would be unhappy. A cell with an exposed telomere will stop dividing. Well, wouldn't that be great if we could make that happen to a cancer cell? Imagine if to a cancer cell, we could stop this process. Cancer cells are rapidly dividing. All the time, they have to have this active telomerase to extend their chromosomes. All the time, this complex mixture of proteins is having to be disassembled so that the DNA can be replicated and then reassembled again on the daughter cells. Cancer cells have very dynamic telomeres. Can we use this fact that whereas most of the cells in our bodies have quiet telomeres that are doing nothing, whereas cancer cells have active telomeres, can we use that as a therapeutic strategy? Can we find a way to destabilize telomeres? Whoop, too fast. Well, that's been the aim of the project that's been ongoing in our lab for quite a long time. We look at this structure and we say, where are the opportunities to interfere with this process? Clearly, there are many targets. There are targets in protein-DNA interactions. There are targets in protein-protein interactions. The one that we're going to talk about particularly is this interaction here between POT1 and single-stranded DNA. And the reason that we focused on this one is because there is crystal structure data for it. We have the crystal structure for the DNA binding domain of POT1. So we have some ideas about how this thing binds to single-stranded DNA, and we can think about the possibility that we design small molecules to actually target this interaction. Because if we can destabilize one or more of these interactions, it's like, as we say, a house of cards, or that game Jenga, yeah? You take one piece out, and the whole thing collapses. And the idea basically with the telomere is the same. We take one of these little pieces out, the sheltering complex collapses, and we get basically typically a cytotoxic event. So this has actually been done previously through a different approach. The original approach for trying to interfere with the POT1 DNA interaction was actually molecules which are designed to do something completely different. The first, if you like, drugs or near drugs in this area are G4 ligands. So the idea between G4 ligands is these are molecules that stabilize G quadruplexes. And typically, G4 quadruplexes, these molecules, are typically small hydrophobic aromatic molecules, typically with positively charged side chains. And what happens to these molecules is they effectively just sit on top of that flat G4 structure. The positively charged side chains go into the negatively charged grooves of the DNA, and that gives it the interaction between the two. So these were known to basically have anti-tumor activity. The question is why? So basically, the idea is that what happens is that in the presence of the G quadruplex ligands, we stabilize this single-stranded region of the DNA in this quadruplex form. Now, the original reason this was done was the thought that, well, if it's basically in a quadruplex form, then telomerase can't work, because telomerase requires an open, single-stranded piece of DNA to act as a template. Remember, it's a reverse transcriptase. It needs something to start with, to hang on to, before it can extend the telomere. If that's not available, because all our single-stranded DNA is scrunched up into a quadruplex form, then telomere elongation can't happen anymore. So that was the original idea behind the discovery of these G quadruplex ligands. And indeed, they work. When you give them to cells, you can see that you inhibit the action of telomerase. But let's think what happens if you inhibit the action of telomerase. Okay, your cancer cell divides, its telomeres get a bit shorter. Now telomerase can't work. Okay, So you've got a cancer cell with a slightly shorter telomere. It's still got a telomere, so presumably it could carry on living. It could divide again. The telomere gets shorter. Okay, but it's still got a telomere. And you can work out that most cancer cells, if you know the length of their telomeric DNA, could actually replicate maybe 20 or 30 times before they would reach, if you like, real danger point. So what you would expect to see when you treat a cell with an G4 ligand would be that you'd interfere with telomerase, but the cancer cell would just divide, maybe 20, 30 times before suddenly you would get a therapeutic response. Now, clearly, that would not be a good thing, because most of our tumors, if they replicated 30 times, well, we'd be dead by the time these drugs would work. But interesting, that isn't what happens. If you treat a cell with a G4 ligand, the tumor cell actually dies quite rapidly. Now, that cannot be because we've just inhibited telomerase. No. The reason pretty much we know that it happens is because we have inhibited the telomeres. Because now, for example, POT1 can no longer bind to the single-stranded region, because it too requires it to be open and extended, not wrapped up in this quadruplex form. So again, the reason that we see a rapid onset of cytotoxic activity with molecules like G4 ligands is because they destabilize the telomeres, not because they're telomerase inhibitors. So this got us thinking, can we think about targeting this process of inhibiting POT1 DNA interactions more directly? These molecules do it by accident. And notice they do it not actually by being a molecule which targets the protein component of the recognition. They do it by targeting the DNA component of the recognition. Can we not think of a much more, simple, straightforward, small molecule therapeutic strategy where our target, instead of being the DNA, is the protein? So that's basically, in words, what I've just said. We know that the theory will work. If we destabilize the telomeres, we can get a therapeutic response, and it's the selectivity there. Can we target the protein component instead? Well, let's go back to that crystal structure data that we have for the POT1 in complex with a piece of DNA. So POT1 DNA binding domain is actually 2 domains. Each one is called an OB fold. It's a well-known domain in structural biology, often associated with binding either nucleic acids or sometimes polysaccharides. So if you like, as a cartoon, I'm just going to draw it as these 2 kind of cylinders. And here's our single-stranded DNA. And you can see the way that it kind of lies across the surface of both of these OB folds. The little lines there, imagine that's each of the little bases in our piece of DNA. So what's interesting, if we look at that a little bit more closely, is that we can see some interesting things. First of all, you can see there's a sort of gap here. There's a sort of cavity between the 2 OB domains. If we look at the way in which the DNA interacts with the protein, there are many complex interactions between the 2, but there's one quite simple, very obvious way, if you like, in which this protein grabs hold of a piece of DNA. And that is that one of the bases in our TTA-GGG repeat, in fact, one of the T's, is sandwiched between 2 tyrosine residues that are actually in the second OB domain. So we have a tyrosine, and one of those T's just sticks nicely between the 2. So in our diagram, we could represent it a bit like that. 2 tyrosines in the second domain, and this T of our TTA-GGG, it's actually that T there, in between the 2. Now, there's this great sort of hole here. So you think to yourself, well, now, that's an interesting little cavity. That looks about the size I could put a drug molecule into. And indeed, when you look at it and you analyze it, and you can do all the sorts of modeling-based things that tell you whether or not a pocket looks druggable, the answer comes back is that that would be a great place to put a small drug molecule. But you can see immediately there's a problem, because I could put a small drug molecule there, but so what? The DNA doesn't go there. Why would I expect that to have any effect on the ability of a piece of DNA to bind to this protein? Clearly, that isn't going to be enough in our molecular design. What we want, though, is we want to make our drug bigger. We want to extend out of this region into this tyrosine sandwich region. Wouldn't it be brilliant if our drug could also have an affinity for those two tyrosines? Because if our drug wanted to sit in there, now we have the possibility of competitive inhibition with our piece of DNA. Now we cannot have the drug in there and the DNA there at the same time. We have a potential inhibitor. So that's the theory. Okay, but before you do that, you probably want to ask yourself, is there any way we could test to see whether or not that is likely to be right? Can we do any target validation on that before we actually go and start making some small molecules? And so we thought that was something that we really needed to do. So in fact, we started, before we made any molecules, with trying to do some, if you like, some biology and screening-based target validation. And for that, we developed a very simple but very useful assay, which is a fluorescence polarization assay. So just to explain how these assays work, the basic idea in a fluorescence polarization assay is that you have a fluorescently labeled ligand, which is bound to your protein of interest, and you attempt to displace it with an inhibitor, or with a competitive inhibitor. The idea is that as long as this fluorescent ligand is bound tightly to the high molecular weight protein, it will tumble slowly in solution. So what that means is that if you shine polarized fluorescent light on this ligand, then when the light emerges again from it through fluorescent, it too is polarized, because the molecule hasn't tumbled very much. But if the ligand gets displaced into solution, it begins to tumble very fast, in which case, even if the light which is absorbed by it is polarized, by the time it is emitted again, it has lost all its polarization. So in a very simple assay, you can tell whether or not a ligand is bound or unbound to your higher molecular weight protein. So this was the idea of our assay. So we had the protein available. We fluorescently labeled one end of a small piece of the telomeric sequence DNA. So the question is, could we find ligands which have an ability to displace? So say the way to work this out, first of all, is to ask yourself, well, is this interaction here with the T important? Remember, that's the one that we're going to displace. But the question is, if we just push that TNA out from between the two Ys, is that going to be enough to make the whole DNA fall away? So we thought, well, one way to do that is to see, let's just not make some drugs, but let's just make some other molecules, some pieces of nucleic acid, small nucleic acid fragments, in which we put something else here at this position X. And let's see if we put something else at this position X, whether or not they can also displace the normal telomeric sequence from the protein. So what do you find? Well, this is just classic binding curves. What do we see? Basically, when we've got the proper specific, if we try to compete off our fluorescent probe with the same sequence, imagine that X is T, but it's unlabeled. Then we've got a simple competition assay. In fact, that's this line here, specific, which is just the one that you can see here. You can see, so the specific DNA containing a T at this position here is most successful at displacing our fluorescent ligand from our assay. Anything else is worse. And that's good, because these other things are, if you like, models for what will happen when we have our drug in place. For example, imagine that we, instead of having a T here, we had an A. Now, it doesn't want to bind to this sequence. It can't displace the fluorescent ligand. Imagine that we remove this base altogether, make a 7A basic sequence. Again, it can't displace the ligand at all. If we just take a completely random sequence of DNA, of course, it's completely unable to displace this sequence from the POT1. So what that tells us is that this T is really important. Anything that doesn't have a T there is not going to be able to displace the ligand. Now, what that means then is that we turn that argument on its head. What this assay has told us is that that T is important. So what this assay tells us is that if we could make a drug which will stop that TNA binding, it should work. So that was our philosophy there. This told us getting that T out from between those two Ys has a good chance of really affecting the ability of this POT1 protein to bind telomeric DNA. So now the question is what on earth should this molecule be? So we did exactly the same sort of protocols that you guys here do when you start off your screening. We actually started off with a fairly focused library because we knew pretty much the sorts of things that we wanted to have in our molecules, or we certainly knew the sorts of things we didn't want. So we could do just using simple QSAR descriptors, 2D pharmacophore models. We were able to reduce the zinc database as it was then to of the order of about 500,000 small molecules to basically put through a docking type screen. So that's what we did. And obviously we were looking for some very particular characteristics. They had to bind, not just bind to POT1, but they had to bind to POT1 in order to occupy this cavity here. And they, very importantly, had to have a functional group or some part of their structure which was going to sandwich nicely into this position here and therefore would prevent the T from binding there. As a result, actually, from our screen, I'm not going to go into the details of all the screening methods. We used a variety of different screens. We tried to come up with consensus views about what was most likely. In the end, you just decide how much money have I got in my pocket today, and you go away and you buy as many molecules as you dare, really. I think that's our process at the moment. I know we've had some interesting discussions with Carlos. I know in a perfect world, that number there would have said 1. And of course, it would have proved to be active. But we don't believe that we're that good at doing this yet. So yeah, we purchased 305. So now what I'm just going to tell you a little bit about is the process through which we basically refined those 305 molecules that we bought. Well, what we actually did is, as I said, the first thing is we had that fluorescence polarization screen. And that was where we had to really test things. And fortunately, the screen is a nice screen. It could do it in quite a high throughput manner. So it was no effort at all to take those 305 molecules through the screen. And, you know, it takes an expert to analyze the results from that. But out of those, we came up with what we reckoned were about 20 decent hits out of that screen. So the question is, well, with these 20 molecules, what were we going to do with those? Well, clearly, there were all sorts of validation that we needed to do. So one of the first things to do was to make sure that our molecules was to go back, if you like, with these 20 at least, to go back to what our modeling, our in silico screening had said about them, and ask, did they actually satisfy the design criteria in the beginning? So I'm just showing 4 of these molecules, because these were in fact the 4 that we focused a lot of our time with. You can see that they're the classic molecules that you'll see when you buy molecules from the zinc database. They have a certain amount of structural things in common, but you could also see quite a lot of structural variability there. But what you can see in all cases when we looked at how it was, if you like, why had we ever decided to buy them in the first place, that they very much did satisfy the design requirements. This is the hydrophobic, or well, actually quite complexly characterized cleft between the 2 OB domains, with lots of opportunities to make interesting hydrogen bonding, other sorts of salt bridge interactions with the protein in this region. Here we have those 2 tyrosines, and here you see the way that both of these 2 ligands, I can't remember which of these 2, I think one of these is HO7, and I think the other one is HO4. Anyway, both of them are, as we required, putting a large, planar, aromatic portion of their structure into this region. So they're doing exactly what we wanted them to do. So that was a good start. Then one of the things that you need to do, obviously, with any assay is wonder, well, are your hits positives really positives? And every type of assay has different ways in which you need to work that out. In the case of a fluorescence polarization assay, there's always a possibility. Remember that the way that the assay works is that when you put your drug in, if it works, you should see a reduction in the fluorescence intensity. That's what should happen. But sometimes you have a molecule which is capable of basically quenching fluorescence directly, not by actually making the molecule dissociate from its target. It just quenches fluorescence. So what you have to do, first of all, with your hits is to see if they're just basically non-specific fluorescence quenchers. And in fact, that's exactly what this assay here shows. So just to explain basically, is the colored bars are molecules really which, the pale bars, this is the apparent activity of the molecules against the enzyme. And what you can see here is that all three of them basically, we've got different amounts of activity, but I mean, some are more active than others, but this seems to correspond more or less to activity. What you expect is when you see if they're fluorescence quenchers is you should not see a signal. And what you can see is, sure, 1, 2, 3, they're absolutely fine. But unfortunately, molecule H7 completely quenches fluorescence. So what that tells us is that although HO7 looked like a really quite potent molecule, it wasn't. It simply happened to quench the fluorescence in the assay. So these are the sorts of important things that one has to be sure of with an assay. You can do an assay, it may be a lovely high throughput quick assay, but do you actually know what the readout is? Do you know what it means? And sometimes, if we hadn't done that and checked that, we could have spent a lot of time trying to understand this molecule HO7 in a lot more detail, and that would have been a complete waste of our time. Yeah, so that got rid of that one. So now we're down to 3 that we quite like the look of. So then the next thing was to ask ourselves, well, this high throughput assay, again, it's a rather indirect way of actually measuring whether or not our ligands are binding. Can we do something else? Is there an equivalent assay? Maybe something a little bit more, well, it is biophysical, or the sorts of assays that biologists are more used to doing. So we went back to an EMSA assay. So I don't know if you know about electron mobility shift assays. It's quite a simple idea. It's a gel-based assay. You have a radio-labeled probe. You have a radio-labeled probe. If the probe is stuck to your protein, then it will migrate slowly through the gel. It will stay near the top. If your radio-labeled probe has fallen off your protein, it'll go fast through your gel, end up down here somewhere. And this is exactly what we see. So this is the probe on its own. This is the probe complexed with the POT1 protein, and you can see that basically it's up here. Now we had HO4, and all our nucleic acid is being removed from the protein. Here we have our molecule DO2. Again, we had the protein, most of the DO2. Here we have our protein, we had our DNA, most of it is up here. We had our DO2, all our ligand, all our DNA is back off the protein. Here's GO2, binds to the protein here. Off we go there. So what that tells us basically is that in all these cases here, we have a situation whereby the ligands are able to more or less, they're definitely removing the protein, they're removing the DNA from the protein. So this assay, if you like, is just confirming what our high throughput screen does, but it's just double checking, because it's always a good idea to have 2 assays for something, not just one, especially if you can have 2 assays which are very different in their character. So again, this obviously is a completely different sort of assay from a fluorescence polarization assay, and that's really useful, giving you confidence that what you're seeing is real. So then we also use this EMSA approach to ask ourselves something about selectivity. We needed counter assays. So we had a very simple counter assay that we set up. We had just hanging around in the lab, because we had some other projects, another DNA binding protein. It's actually the SSB protein from single-stranded DNA binding protein from B. subtilis, and it's a very non-specific single-stranded DNA binding protein. It has the same OB domains, so it has some similarity to POT1. But unlike POT1, it'll bind pretty much any piece of single-stranded DNA. So now we ask the question, are our ligands that we've made, are they really working selectively? Are they selectively removing DNA from POT1? Or are they the sorts of molecules that would remove any sort of DNA from any sort of DNA protein complex? Of course, that would not really be very exciting. So this is the assay. We run it in exactly the same way as the EMSA assay, only of course, instead of using the POT1 protein, we use this bacterial SSB protein. So this is what we expect to see. Here's our radio-labeled oligonucleotide, and as we titrate in more SSB, it all basically complexes, soaks up like a sponge, it soaks up our DNA, which is exactly what we expect to see. We know that the SSB protein will bind any DNA, and TTA-GGG is fine as far as it's concerned. So there it is. So then the question then is what happens to that affinity between the SSB and our DNA in the presence of our ligands? So what do we see here? We see that basically as we increase the concentration of HO4, we displace the radio-labeled DNA off SSB. But it doesn't matter how much DO2 we add or how much GO2 we add, we almost never or almost never can compete the DNA off the SSB. So what does that tell us? That tells us basically that this HO4 is not really actually an interesting molecule because it's non-specific. It has the ability to displace telomeric DNA from POT1, but actually it can displace any DNA apparently from, or maybe could displace any DNA from any protein complex. So that's hardly going to be a recipe for a successful drug. We need selectivity. So again, at this point, we could abandon HO4 and fall back on our two favorite molecules, DO2 and GO2, as having passed this next level of, if you like, validation. So then, if you like, we try to do something even more biological. So this is, again, just showing examples of one of these molecules. This is DO2. We start going into cells. Nothing I've shown you so far has really been in cells. So we have to ask ourselves, what are the sorts of activity that we expect to see in a cell if we are hitting the target that we want to do? And of course, this is always one of the most difficult things when you go from an in vitro to a sort of in vivo assay. Is your readout representative of you hitting the targets you think you're hitting? And of course, that's a very difficult thing to do. But what do you think about is, well, what do you expect to see, obviously? Well, what we knew is that if our molecules were actually in cellules disrupting telomeres, then what happens when a telomere gets disrupted is you get a DNA damage response. And sure enough, what you can see is that our molecules induce a DNA damage response. So this is gamma H2AX phosphorylation, classic DNA damage assay. Here's our control. Here's irinotecan as a positive control. And you can see that DO2 will give basically as much DNA damage as irinotecan. So that suggests that that's one thing that we thought that these molecules ought to do, and they do. Are they basically cytotoxic? Classic MTT assay. Again, this is all in HeLa cells. And again, you can see that these molecules have, micromolar. No, they're not very potent, but they have a respectable, maybe somewhere just under 100 micromolar, considering it's basically just a hit. 100 micromolar activity against HeLa cells. They have the cytotoxic activity that we would hope for. And again, what you normally expect if you disrupt a cell, if you're going to get this cytotoxic activity, then you're going to hopefully see all the markers of apoptosis. And indeed, here's just there's an annexin-FITC assay, just looking basically for induction of apoptosis by these compounds. And again, so the controls are just the light bars and the treated are the dark. And you can see a time and, in fact, a dose-dependent response there to treatment with these cells. So this is all wonderful evidence that they could be working by the mechanism that we say. But you'll notice that these panels are marked B, D, and C. So what happened to A? Well, of course, A is the one that this is where we haven't got the assay that we really want. What we're always looking for in these things is what is often called the smoking gun, yeah? The proof, almost beyond doubt, that your molecule is working the way that you said it was, the evidence you can't dispute. And if there would be one piece of evidence that would have been lovely to have seen with these molecules, that would have really shown that these events must be things that are happening at the telomeres rather than anywhere else, would have been if we'd seen basically what are called chromosome fusions. Because what happens is if you disrupt the structure of sheltering at the end of a chromosome, is you expose the very tip of that chromosome as a piece of naked DNA, and it's got an end. And the cellular repair machinery will see that, well, as DNA damage. And what the cellular repair machinery normally does when it sees an end of a piece of DNA is it looks around in the cell for another end of a piece of DNA, and it joins them together in the hope that those two things belong to each other. So what happens in the case of disrupting telomeres, if you do it right, is that in fact one chromosome and another chromosome end up fused together through their exposed tips of their DNA. So the smoking gun for us would have been to see basically bridged chromosomes, fused chromosomes in metaphase spreads, and rats, but at the moment we can't see them. We've got all this indirect evidence, but the thing that we really wanted to see, it's tough. These experiments are, certainly in our hands, this is not our area of great expertise, but we tried very hard. But so far we've not been able to show the real smoking gun, which would be chromosome fusions. So we fall back, we have lots of more indirect assays, though, that are looking hopeful. For example, we can make cell lines that overexpress POT1. These cell lines that have more of this protein around become resistant to the activity of our drug, which is what you expect, because basically this excess POT1 can soak up the drug, so it can't then interfere with the DNA binding process. So that's good. That suggests they're working by targeting POT1 in a cellular environment. But that's, if you like, basically where we are at the moment. We've got small molecules that without a doubt have the ability to displace single-stranded DNA from telomeric POT1. We've got some very selective molecules, some are less so. We've got all sorts of interesting biological activity in cells that's consistent with destabilization of telomeres as being the way they work. And we've got overexpressing POT1 cells showing resistance. So all of these things are really positive things that we've got molecules working the way that we want. But this isn't enough right now. We need more. Certainly in the environment where I work, we cannot progress until we've answered some more questions. We need to demonstrate telomere disruption more directly. There's some other work we need to do, which is not difficult. We need to look at other cell lines. We need to make sure that this is something that works in other cell lines. Very importantly, you'll notice I've described no chemistry in this program so far whatsoever. Everything has been done with molecules that you can buy in. And this can get you so far, but what was interesting is that we hope to be able to do what's called SAR by catalog, which is basically whereby you've got a hit from your screen. You know that you only screened a tiny subset, 20 or 30 molecules out of your database because you couldn't afford to. What you do is you go back to that database, you say, well, there must be a few other molecules like this that I can also now buy, and I'll screen those. And then I don't have to do any chemistry, but at least I'll get some idea if there's a structure-activity relationship. Unfortunately, when we go back to our catalogue, we can't find any molecules really that are close enough in structure to our hits that we could use them for any sensible SAR-type activities. They're all kind of singletons. Now, sometimes that's good, because what it means is that you've got a scaffold which is quite unique, where if you did do chemistry around it, you would have a chance of making something very novel. And as we all know, if you don't have novelty, you're hiding to nothing in terms of trying to develop new agents. And that's the good news, but it means basically that you need chemistry in-house to do it. Now here at Nottingham, we have chemists to do it, but of course you can't get that done until you can get the money to do it. And before we can get the money to do it, we have to answer some of these questions up here as well. So for the moment, our questions are mainly around improving our biology. Then hopefully we can move into the chemistry to answer these questions down here. So as I say, it's an incomplete piece of work, but I just wanted to demonstrate the way, if you like, in which with a good team of good collaborators with lots of different areas of specialism, you can really do a lot in this area, but you need that. You cannot do these things alone. So I've had a great group of postdocs and PhD students in the lab over several years, all working on aspects of this project. And I can't say any one did more than the others. All of these people contributed to the work that you've seen here. I would not have been able to do it. I say my background is in medicinal chemistry and modeling. Without my good collaborators, Ludo Decker, who's a great screening and pharmacologist specialist, Panos Sultanas, who's an expert in DNA protein recognition, and a collaborator of ours, Mark Hummerson, in a small pharmaceutical company that was associated with this project at one stage as well, who provided a lot of very valuable med chem insight into what we were trying to do. And again, we would not have been possible to do any of this work without funding from many different bodies in the UK. So with that, thank you very much indeed. Again, it's been a pleasure to be here. I'm sorry, I think I spoke for too long. I apologize for that. Thank you very much. If there are any questions, of course, I'd be very happy to try and answer them. But anyway, my main thing is to say congratulations to you all and best luck for the next 10 years. And I hope we can be part of that with you. Thank you.
So, I wonder if there have been any studies in this coordinate sequence, if the damages to any of those bases would interfere in the binding of the...
There's a limited amount of evidence for that. I mean, the specificity of these proteins obviously is very high. It has to be in order to cause the complex to assemble. So there's a fair bit known about what sort of can be allowed and what is not allowed in that. And that's very interesting from a pure science perspective, and in fact we've done some work on that. Of course, it's difficult to see how that could be a therapeutic strategy because, well, unless you've got a brilliant idea for it, because it's a question of how one would induce a particular piece of damage to your DNA just in that region, you see. It would be quite non-specific. I mean, if you could think of a way of doing it, I agree, it might be really interesting. You know, perhaps if you could think of some way of a damaging agent that was selective for TTA-GGG, yeah? Then, I mean, I could imagine that could work. So if you ever hear of one, please let us know.